Pleasure thank you. It's my pleasure to introduce Sam Clamens. Sam got his degree at the College of William and Mary in mathematics and computer science, so mathematics with a minor in computer science, and came to Caltech in bioengineering. He's going to tell us today about some of the work he's done here as a graduate student. But I also like to just say that in addition to what he's going to tell us about today, Sam's done a large number of other things kind of in the spirit of community science. So he was very involved with iGEM, I think, at College of William and Mary, but also when he was here, he helped out a group at a high school, Alverno Heights Academy, and was very active in getting a group of high school students to think about synthetic biology. He also played a leading role in putting out a very large collection of parts that we had from our lab and putting that out to the synthetic biology community and making it available to everybody else. So in addition to the work that he's done and the things that people will build on based on what he's done, I think one of the things that Sam has done is to really make contributions to the community at large, in addition to all the things he's done in our lab and other things. Hey, Paul, welcome. So it's with great pleasure then I turn it over to Sam to tell us a little bit about some of his efforts and his PhD thesis work. Sam, over to you. You're muted.
There we go. So you should now be able to see what we're going to talk about. Yeah, this is the content of what I'm going to talk about. So I'm going to start by explaining just what I mean by dynamic CRISPRi circuits and where this falls in the space of larger synthetic biology. And then I'm going to focus in on a very specific synthetic biocircuit that we can make with CRISPRi and talk about how we can understand it and how we can make it better. So first, to give you some context, what is it that we do? We are a synthetic biology lab in the Murray lab. We, in the briefest possible terms, build interesting devices that can sense, respond to, and record information from the environment. But those devices, instead of being built out of silicon and wires, are built out of living materials like E. coli, primarily E. coli. And we can program E. coli as living devices in a number of different ways. But there's really one fundamental technology that forms the basis for everything I'm going to talk about. And that's the repressor. So let me start by just, for those of you who don't know, explaining what a repressor is. So everything we build starts with some piece of DNA. There's some DNA. On this DNA, you have genes that can express a variety of different things. Importantly, every gene comes with a promoter, which is a piece of DNA that tells the cell this is something that can be transcribed and turned into, ultimately, a protein. And protein could be a sensor, it could be a fluorescent reporter, it could be something that goes and acts on other genes, it could be any number of things. Now, a repressor is a protein also, which has the important property that it can bind to a specific sequence on or around the promoter, which shuts down production of the gene associated with that promoter. So a repressor is essentially the biological equivalent of a NOT gate. If the repressor is there, you don't make the downstream protein. If it is there, if the repressor is there, you do not make the downstream protein. If the repressor is absent, you do make the downstream protein. This is important because you don't usually want proteins to be on all the time. You want them to be on conditionally, depending on either something in the environment or something about the state of the cell. Now, also critically, repressors are proteins. They come from genes. Those genes have promoters of their own. And the things they target can be repressors themselves. So you can repress repressors with repressors, and you can chain these together in almost arbitrarily complex ways to make complex networks that do interesting things. And we usually write that down kind of like this. We draw it as the orange thing represses the purple thing with an arrow with a blunt end to say that that's what the relationship between them is. So let me describe a couple of really classic examples of repressor-based synthetic biocircuits before we get to the real meat of this. One example is the toggle switch, which comes kind of from the idea of making a biological equivalent of RAM. So if you want to make an active but persistent memory that can have different states, how would you do that? Now, in early electronics, one way you could do that is with a logic gate called a flip-flop or something similar, which is a little conglomerate of logic gates that can be set so that one but not both of the two outputs is high or on. And once that's set, it will stay that way until reset by an active signal to potentially flip it. And there's a biological equivalent of this, which is called the toggle switch, which is simply two repressors that repress each other. So if you think about this, if A is on, if there's lots of A present, that will repress B. B can't be produced, which means A is allowed to be produced and it's sort of stuck, it's stable. And vice versa, if B is on, then A will be off, which means B can be on. So does that device actually work? I've made some claims with some English sentences about what this should do. If we want to actually check, we should do some math or some experiments, but math is easier. So we can do that with phase plane analysis. This is a simple enough circuit that we can do this. So we can look at the space of all possible combinations of concentrations of A and B. So each point here is one state of this two protein system. And we can draw something called the B nullcline, which is the set of all combinations of A and B where B doesn't change, where B is stable. And another way to think about this is if you could fix a concentration of A, any concentration of A, the B nullcline gives the concentration of B that the system would settle to after a while. And similarly, you can draw an A nullcline, which is the same thing, but for A. If you set a concentration of B, the A nullcline says what A will go to after it stabilizes. And you'll notice that there's only one point where these two nullclines cross, which means there's only one point at which the system is overall stable, where both A and B don't change. And if you look at what the system does, no matter where you start, it will always flow to this one stable steady state, which means this is not a toggle switch. Why? Because there's only one thing it can do. It doesn't have two possible states. You can't flip between two possible states if there's only one thing it can be. Fortunately, for synthetic biology, real repressors don't have nullclines that look like this. They don't have binding responses like that. They actually look more like this on the right. So if you add A to the system, you'll find that B at first doesn't change very much. Adding a little bit of A does very little, but once you cross some threshold of enough A to matter, you get a very, very fast response in B. And then after that, adding more A doesn't really change B by very much. And you get a similar dynamic for A. A little bit of A does not much, not much, not much. A moderate amount will repress by a lot, and more after that doesn't repress much more. And the upshot is you end up with a phase plane with three intersections of these nullclines, which means three places where the system is stable and steady, or sorry, where it is at steady state. You find that two of these steady states are stable. The one in the middle is not. So if you start any distance away from this center steady state, it will flow to one of the two stable steady states, which represent the two toggle positions, one where A is high and B is low, one where B is high and A is low. So this describes an effective toggle switch. So hopefully this gives you an idea of the kind of analysis you can do on a sufficiently simple biocircuit. I also want to introduce this S-shapedness of the binding response of A and B is something that we generally call cooperativity. You can get it chemically in a number of different ways, but mathematically it expresses itself as this, again, this S-shaped response curve. Okay, and you need that for the toggle switch to work. If you don't have that, you get something like this. You have no toggle. Here's another example that we're going to talk about a lot for the rest of this presentation. This is the repressolator, which is very similar to the toggle switch, but it has three repressors instead of two. So if you think about what this does, if you start with a lot of A high, that will repress B. It will cause it to not be expressed. You won't have much, which means C can be turned on. It will be high. But if there's a lot of C, that represses A, so A will go low, and now B can go high because it's not being repressed, and that forces C low. So what you end up seeing over time is an oscillation. So this is a real repressolator built so that each node of the repressolator is associated with a fluorescent protein of a different color. And what you'll see is these cells are oscillating between colors, where they're going from one of these being high to the other being high to the other being high, and so on. And interestingly, in the classical original analysis of the repressolator, you find that this circuit also requires some cooperativity, just like the toggle switch. There's this shared sort of structural requirement between the two. So those are a couple examples of simple biocircuits of the kind that we might build. But in practice, in principle, we can scale these up and we can make many different kinds of circuits. We can make them various sizes, but in practice, we're limited by a number of things, but principally the number of possible repressors you can use at once. So the largest set of repressors that are kind of functionally verified to all work together is this set on the left produced by Chris Voigt's lab that's somewhere between 12 and 16 repressors, depending on how you count. And the Voigt lab has some really nice software called Cello that can take small logic circuits and compile them into DNA that use these repressors to implement the logic circuit, so that they take some chemical input or some upstream gene input and turn it into expression or not of some downstream gene, depending on the logic gate. But this took a lot of work to make, and it required combinatorial work. So the Voigt lab had to characterize not just every repressor in this system, but every pair of repressors. So they need n squared work to get more repressors added. And if we want to make something complex like a cell, we're going to need a lot more repressors than 12. This is the core transcriptional network of E. coli. It's small enough that you can write it down, but it is a lot more than 12 pieces. So we'd like to be able to make repressor-based synthetic networks that are larger than a dozen pieces. And this is kind of the inspiration for bringing on a technology called CRISPRi. This is a programmable repressor. It's made of two different parts, an RNA called a guide RNA that has a constant structural region that makes it a guide RNA, and a variable region that acts as a sort of targeting sequence. And an enzyme called dCas9. This is a deactivated version of the Cas9 endonuclease that some of you may have heard of. The important thing is that it will bind to guide RNAs, and those bound complexes will bind to their targets depending on the sequence of the guide RNA. So it acts as a repressor. It will take a promoter that normally would transcribe, it will bind to it, and it will inactivate it. And importantly, guide RNAs, of course, are produced, much like any other RNA species, off of promoters, so they can be targets. So this effectively gives you a way to repress guide RNAs with other guide RNAs. So again, CRISPRi is a programmable repressor. By expressing different guide RNAs, it becomes a repressor that targets different sequences. And the only thing you need to change to make it target those different sequences is the sequence of the RNA itself. And we expect that they're on the order of single digits to tens of millions of possible guide RNAs that you could use in principle in the same cell. So this is perhaps an extremely scalable version of a repressor. But there are some limits to CRISPRi, and we want to try to understand those limits and make sure that CRISPRi actually can be used to build complex devices that we may care about. And particularly, I want to call out that the CRISPRi system doesn't have any of the chemical quirks that would give it what we would call cooperativity. It doesn't have this S-shaped binding curve. So already we might suspect, writing this down, that it may not be capable of doing everything we'd like from a repressor system. So we'll start trying to understand what CRISPRi can do by looking at a really, really oversimplified model of CRISPRi. And this is an equilibrium model where we assume that guide RNAs and dCas bind very tightly, that complex binds to DNA very tightly, that you have infinite amounts of dCas at your disposal, and that binding happens instantaneously. This is not particularly realistic, but already it'll give you a binding curve that looks like this. This is very unlike the S-shaped binding curves that you see from standard repressors. It's basically piecewise linear. It's not quite, there's a little kink right at the corner, but it's essentially piecewise linear. And we can use that to get some understanding of what you can do with it. In particular, you can make a toggle switch from this oversimplified version of the CRISPRi system. And if you do the same kind of phase plan analysis that I briefly showed earlier, you can see that you can get a toggle switch that has 3 steady states, 2 of which are stable at the outsides, and 1 of which is unstable and sends flow of the system to those other ones. So if certain parameters, conditions are met, a toggle switch made from CRISPRi does function. That's already a little surprising because classically toggle switches are understood to require bistability. What we've shown is you don't need that. Just a totally linear binding curve with a fast saturation once DNA is completely bound out is sufficient for the CRISPRi toggle to work. And you can do the same thing with the toggle switch. You can use this to show that, again, under the right parameter conditions, you can expect a CRISPRi repressilator to work. Again, surprising. Typically, we think that requires cooperativity. Turns out it might not. But this really is too simple of a model. This is really just to verify that you don't necessarily need cooperativity to get interesting circuits, and that it might be worth pursuing CRISPRi. But there are some important limitations of that really simple model. Particularly, it doesn't really have kinetics. It kind of does, but it doesn't have the stepwise kinetics that real CRISPRi has. In real cells, Cas9 is limited. There aren't infinite amounts of Cas9 molecules, and every guide RNA that's added adds competition for that against all the other ones. So like from the literature, we've seen that as you add guide RNAs to a CRISPRi system, the effectiveness of those guide RNAs drops pretty precipitously. And we want to be able to see that in our simulations and understand how that affects circuits. So let me describe a more complete model, and this will look a little familiar. Again, we start with guide RNAs and Cas9, which can bind and unbind, though in practice they don't really unbind very much. These can bind to promoters, which otherwise would be producing things, and once they're bound, they don't produce much. This model is going to need dilution. So in bacteria, in E. coli, the primary way that the cell gets rid of things is through growing and just diluting out components. It doesn't really destroy most things. It'll just grow and not replace them. And that's represented by all of these dilution reactions to get rid of things. And in particular, I want to call your attention to this particular dilution reaction here. It turns out that dCas9 binds extremely strongly to its targets. And the only thing that really removes it from DNA is when that DNA replicates, which happens at the rate of growth so that you have balanced amounts of DNA, balanced production of DNA. So we're going to have dCas9 unbind at a rate of dilution. That'll set the time scale of a lot of these simulations. Notably, guide RNAs are produced from promoters that can be bound in this model. dCas9 doesn't. dCas9 will come from nowhere. We don't really care where it comes from, but it's produced at a rate. And we'll also optionally allow a leak from bound promoters so that a bound promoter might be able to produce a little bit, even if it shouldn't really be producing anything. And we can get parameters for this model from the literature. Some of these are generic estimated rates of RNA and protein production or cell growth, as down here. We can get binding parameters from a variety of in vitro and in vivo experiments that have been done to specifically characterize this enzyme. I do want to call attention to the fact that dCas9 doesn't really let go of anything it attaches to. Functionally speaking, it is slow enough at unbinding from guide RNAs that it essentially has no unbinding. And that complex effectively doesn't unbind from DNA, except through dilution, which is modeled here as a separate reaction. Now, I'm going to talk a lot about using this model to model the CRISPRi repressilator or the CRISPRilator for a few reasons. Repressilators are really well understood. They're a really classic biocircuit. It's not totally new territory, so we know what we might expect to see from a more traditional repressilator. And we can see how a CRISPRilator differs from that. Repressilators are dynamic. So most work done with CRISPRi systems to date has been with endpoint circuits like logic gates, where you give some input and then you wait for a while and you come back and you see what it has settled to. But we want to actually find out if you can build dynamic circuits that change over time and respond actively. Repressilators occupy a nice space where they're simple enough to look at and understand relatively easily, but they're not trivial. They have multiple different operating regimes, depending on what the parameters of your system are. So they're not totally uninteresting. And they have this structural requirement for cooperativity, or at least classically they're understood to have this requirement that's common across a fairly large range of circuits. So by understanding repressor we get a little bit of insight into what CRISPRi can do in terms of building a fairly large class of biocircuits. Now, I'll skip right to the punchline now. If you use this model to simulate 3 guide RNAs repressing each other in this circuit, you find that it does oscillate, just like you would expect a repressor to oscillate. But it's a fragile repressor. Turns out there are a lot of parameters that you can shift just a bit that will destroy oscillations. So you can either add or remove decas from the system, and it will stop oscillations from happening. Some strange things like increasing the binding rate between guide RNA and Cas9 will kill oscillations in the system. And perhaps most worryingly, even a little bit of leak will destroy this. So again, this is the rate at which a bound promoter will still be able to transcribe even though it's repressed. And even a very, very small amount of that completely destroys oscillations. And I would argue that this is actually pretty well represented in experimental findings. So in the time since I started doing this work, 2 different labs have built CRISPRi repressolators, shown in top and bottom. And what you can see is that they do repressolate. They do oscillate in an order, but it's really messy. They have highly variable amplitudes. They have highly variable frequencies. They don't really go to 0. They don't go to the same baseline all the time. They seem to be working, but poorly, which may be accurately reflected in this model. So we can now ask, using the model of CRISPRi we have, are there interventions we can use to make the repressolator more robust, more stable? One that I found kind of surprising was if you add nodes to the repressolator and you just make it bigger, it turns out to be more robust to a number of perturbations. Shown here, if you add a little bit of leak, destroys the 3-node oscillator, but not the 5-node oscillator. So in some cases, bigger is in fact better and not just harder. As another example, you can add Dcas9 degradation to compensate for leak. So again, as I said, most components of the cell are removed by dilution, not by any active destruction, especially in prokaryotes. But you can add a little protein tag to an enzyme like Dcas that will cause the cell to label it as malformed, essentially, and destroy it actively. And what this simulation is showing is that if you do that, you, 1, speed up the time scale of oscillations quite a bit, and 2, you make your circuit much more robust against leak. Now, I've made a sort of another English sentence claim about this compensation, and I'd like to study that a little more systematically. And to do that, I'm going to show you the results of a lot of simulations, many, many simulations. And what I don't want to do is sit for days and days at a time, running simulations one at a time, looking at the results and saying, this oscillates, writing down on a piece of paper. No, we want to automatically detect oscillations in simulations. And we could try to do this, and traditionally for a repressolator, we might do this, by finding the steady state of the oscillator system, which will always exist. It might be a stable steady state, which you can calculate algebraically. And if it is stable, then this repressolator will always sort of drag the state to that point and stay there, and you will get no oscillations. Where if it's unstable, then the repressolator can never go to that point. It will always kind of oscillate around it. And you can do this kind of analysis on a standard repressor-based repressolator, and it's a pretty good way to know whether there are oscillations or not. But this turns out to not work for a CRISPRi oscillator. And the reason is you can get an unstable limit cycle within the stable limit cycle of the repressolator, which is new behavior. So up in these top 2 plots, I'm showing that exactly the same repressolator can give different behavior depending on the initial condition. So if you give it enough of a kick by starting with lots of one guide RNA, then it will oscillate exactly as you'd expect a repressolator to. But if you start with more even amounts of guide RNA, even slightly more even, it will instead decay down to just a fixed, uninteresting steady state. And if you plot the behavior of a bunch of different instantiations with different initial conditions in sort of the space of all three of the guide RNAs, you'll find that there's this unstable limit cycle. There's a boundary, this red line, where if you start inside that line, roughly speaking, this repressolator will just spiral into a fixed point and stay there, and it will not oscillate. And if you can escape it, then you instead get attracted to a stable limit cycle, which is this blue curve outside, and give you repressylation. So that's interesting in the sense that CRISPRi oscillators actually give you a behavior that you don't get from a standard repressolator. That's kind of neat. The unfortunate upshot to this is that just looking at the stability of the steady state doesn't tell you if you can get oscillations, because in this case, it's stable, but you can still get oscillations. So instead, we're going to have to find oscillations numerically, kind of the dumb way, where we just look for places where a guide RNA goes from increasing to decreasing, and we'll find those peaks over time until they converge to some height. And we can look at the distance between that peak and the last trough, where the guide RNA was lowest. And if that distance is very large, we can say confidently oscillations are happening. If that gap is very small, we can say that it is converged to or is converging to a steady state where there are not oscillations. And if it's an intermediate, then we can print out a graph like this and have a human decide whether it looks like oscillations or not. And if we do this, we can get a kind of specification sheet for a CRISPRilator in the space of two different parameters. So blue dots in these graphs are combinations of parameters for which the repressolator oscillates, and red dots are combinations of parameters that do not oscillate. So on the y-axis, we have the leak rate. So again, this is the amount of expression from a bound promoter that you get as a fraction of the expression you get from that promoter if it were not bound. And this is a parameter that we can't really control. It's characteristic to the Cas9 CRISPRi system and is hard to change. And we don't really know its value, except for some bounds that we know it's probably not more than somewhere between 10% and a few percent. So there's a bound somewhere in this range for where we think it actually is. And the x-axis is the rate of degradation of Cas9 in units of dilution. So all the way on the left is 0 degradation. The only removal of Cas9 over there is dilution. And the 1x is where degradation happens that is as fast as dilution. So that's sort of twice as much removal of dCas9 as you get just from dilution alone. And we can see a couple of things from these spec sheets. One is that the 5-node oscillator does seem to be more robust than the 3-node oscillator. It operates in a greater variety of range, greater range of parameters. We also see that adding degradation does seem to compensate for leak up until some point where when you add too much degradation, you fall off a cliff and oscillations stop working at all. This is important if you were going to try actually adding degradation, because it turns out depending on what degradation system you're using, you might or might not fall off that cliff. So the most common protein degradation tag we use in E. coli synthetic biology is SSRA. And it looks like that tag is going to be far too strong. It's just going to completely destroy oscillations if you try to use it in a cell. There's an alternate system called the MF-lon protease system, which looks like, yeah, if you pick a weak enough MF-lon tag, you might be able to make a CRISPR oscillator that is more robust. Now, I've already shown you that there exist real instantiations of the 3-node CRISPR oscillator, and notably, none of them added degradation tags to their Cas9, and yet they oscillate. Maybe weakly, maybe messily, but they do oscillate, which suggests that if this model is reasonably accurate, we have to be down here in this corner where there's no degradation and almost no leak. that's kind of interesting because, again, the only experimental bound we have on leak is that it's not more than a few to 10% up here. I will claim that what we may be learning from this is that CRISPRi has essentially no leak at all, in the truest sense of leak. So if you experimentally try to characterize CRISPRi by turning on CRISPRi and turning it off and looking at the expression of some downstream thing that is or isn't being repressed, you'll see that when it's repressed, it produces maybe 1% of the amount that it would when not repressed. I'm going to claim that that may not be coming from bound promoters, that actually represents entirely expression from promoters that just haven't been bound yet, that either were recently replicated or have been kind of unlucky in the sense of not being bound by a repressor, but that sort of true leak in the sense of transcription happening through a bound CRISPRi, a bound DCAS molecule, doesn't really happen. Now, you might also look at this real data and say, well, that doesn't look all that much like what I've shown in simulations. It looks much messier, much less smooth. And there might be intrinsic complex mechanistic reasons for this, or it may just be that the models that I've shown so far haven't accounted for simple run-of-the-mill molecular fluctuations. So everything I've shown so far has been with an ordinary differential equation model, which implicitly assumes that everything happens at high concentrations, at large volumes, so that molecular fluctuations average out and give you really nice, smooth, clean dynamics. But real cells don't necessarily meet those conditions. In a real cell, there might only be 10 or 100 Cas9 molecules, and those molecules aren't going to react at exactly what their rate constants would predict. They're going to bump randomly around the cell until they happen to hit their targets, and then they might or might not actually react and undergo chemical reactions. So in real cells, dynamics are stochastic and there are random fluctuations. So maybe that's the only real difference between our simulations and what is seen in real cells. If so, we should try simulating this same system, but with stochastic noise and see if it matches more closely. Classically, that's a little harder to do. Stochastic simulations are more computationally intensive, but fortunately I have access to a really nice package called bioscrape made by other members of the lab that lets you specify a chemical reaction system or a synthetic biocircuit programmatically, and then simulate, and with one Boolean switch, you can flip from doing a deterministic simulation to a stochastic system with molecular noise. So we're going to do that, and we're not just going to do stochastic simulations, but we're going to do lineage simulations. So what's that? Well, biocircuits don't work in fixed size reactors with inflows and outflows. They happen in cells that grow and divide. And this can be really important because when a cell divides, it has to divide up its contents into the two daughter cells. And that can happen in a variety of different ways. So a division doesn't just divide its contents one way. A division event really describes a probability distribution over possible daughter cell states, some of which could be wildly different from the parent. And these all happen with some probability, and we want to model that. So a lineage simulation is one where the reactions that we're going to simulate happen in a cell which grows in volume as it creates molecules. And then once it reaches some critical size, it will divide into two cells where the contents of those cells are randomly partitioned from the contents of the original cell. Now, this is going to take us on a bit of a digression into another chapter of my thesis, which popped out while doing this work, which comes from a problem with lineage simulations when you start simulating DNA. Now, a little background. Typically, when we model synthetic biocircuits or other biocircuits, we tend to assume that DNA is a fixed quantity. It's not really a species so much as a parameter that's fixed by the cell, because we tend to assume that cells hold DNA fixed at whatever concentration they're at. Now, the first thing you might try to do in a lineage simulation would be to translate this idea and say, all right, each cell has some copy number of DNA, and that copy number stays fixed as the cell grows. And then when the cell divides, each daughter cell gets that same copy number. So the DNA of the parent is copied instantly at division time. But this actually gives you some really wild behavior that isn't realistic or particularly desirable, because a fixed copy number in a growing cell means that the concentration of DNA in that cell oscillates in a sawtooth manner. So as the cell grows, the concentration of DNA in that cell drops if you're not adding more DNA, which means reactions with that DNA become less likely to happen and they slow down. And then when you divide, you're instantly injecting effectively a bunch of DNA, and the concentration jumps up, and you get discontinuities in behavior that aren't really realistic. And the reason they're not realistic is because real cells don't keep a specific fixed copy that instantly doubles right before division. They replicate their DNA over time. So in something like a plasmid, plasmids will be produced one or two or a few at a time to balance out the concentration of that plasmid as the cell grows. We want to add that to our lineage simulation. We could do that sort of an obvious way by just adding a reaction that says DNA can replicate. So one DNA can kind of split to become two DNA. But if you write that down and you try simulating a CRISPR later, you get something really wild that doesn't seem to oscillate in a nice way at all. And it gets much worse very quickly over time. And it turns out the reason for that is that the DNA of the different parts of this oscillator is going crazy. It's not staying stable, it's random walking. And it turns out that's because this sort of trivial replication mechanism isn't stable. If you look at simulations of just DNA doing nothing else except replicating and diluting, according to this mechanism, these lines represent different simulations starting with different initial conditions, different initial amounts of DNA. And what you find is they're not stable at all. They random walk, at random, and will eventually either die out or explode to some really, really high concentration that your computer won't want to simulate and will crash on. So why? What's going on here? Well, if you write down the mathematical dynamics of what's going on, you get something like this, where the rate of production of DNA is proportional to, it's equal to some amount proportional to the amount of DNA that's present minus the dilution of that same DNA. And at steady state, this gives you the condition that production must be balanced with dilution, which makes sense. You have to be producing at the same rate that you're diluting. Otherwise, you either get infinite amounts or you go to zero. But there's no bound on DNA. It turns out this is a neutrally stable system. So if you think about this another way, if you add a few pieces of DNA that are replicating trivially, now there's more DNA that can replicate, and the overall rate of production of DNA has gone up. So now you pushed towards making more DNA, having made more DNA. And conversely, if you remove a few pieces of DNA, now the overall rate of production of DNA has dropped, and you're more likely to go to lower amounts of DNA. That's not stable at all and isn't realistic. What we'd really like to get balanced production of DNA that doesn't go crazy like this would be to have replication be not dependent on the amount of DNA present, but just happen at a constant rate. And you can get this with several different ways, but here's one way that I'd propose is pretty good and simple. So in this system that I call dummy triggered replication, you make a sort of dummy replication molecule R at some constant rate, and the cell just makes this out of the ether. And R molecules react with DNA to make them replicate. So what this mechanism effectively does is it decouples the rate at which replication happens from the actual mechanism of replication. And if you simulate this, you find that whatever your starting concentration of DNA, it will pretty quickly converge to a nice steady state, and it'll fluctuate around that steady state. Now, I don't have time to go through the details, but I'm going to claim that this isn't just a nice, convenient hack that kind of works, but there's actually some biological plausibility to this dummy triggered replication model. All the way on the left, I'm showing a model description of the mechanism that the Col E1 plasmid uses to replicate. And what I've shown in my thesis is that with a series of simplifications and with the right simplifying assumptions, you find that this system gives you a replication rate that looks like this. It's a combination of parameters that has no dependence on the amount of DNA present, which is effectively equivalent to this zero order production. And to be clear, I don't want to claim that cells actually replicate using dummy triggered replication, but more that there's a space of models where more complex models can achieve better realism. And there's a smooth simplification you can make from the Coley 1 model down to the dummy triggered replication model. And this is a good enough model to be useful. And other kinds of DNA might use different replication mechanisms. But I suspect, I haven't shown, but I suspect that if you aggressively enough simplify these down, you'll get something close where you'll find a zero order replication mechanism sort of buried at the heart of each of those replication mechanisms. And just to drive home this point, I'll also claim that trivial replication of the kind that I showed before is way down here in the corner, where it's very simple, but also not realistic at all, and you should not use it. You should use something like this, or if you don't trust it, use a more complex, more realistic model. So all that being said, we now have the mechanisms required to simulate a stochastic CRISPR oscillator. So here is a 5-node CRISPR oscillator starting from a single cell that grows and divides into something like 128 cells over time. Now, with a stochastic simulation, we can ask some interesting questions that we couldn't before, like, does a CRISPR stay coherent over time? If you have a population of cells that start in one CRISPR state, will they oscillate together or will they drift? The answer is they do drift somewhat, and if you let them go long enough, they will decohere entirely. But over a period of days, they actually oscillate reasonably in sync. And again, I think we've seen this pretty well from actual repressor experiments. We can also, the whole point of this was to compare it to real data, so we can do that. And I will claim that the stochastic CRISPR can get us fairly close to some, but not all of the experimental results that have been found in the field. So these two, this is a simulation using my model, using the dummy triggered replication of plasmids. It's scaled on the x-axis to be the same time scale as this real data down below. And you can see there's quite a bit of difference, especially in the speed of oscillation of these two. And the cells that were used for the real experiment here were fairly slow growing, which accounts for some, but not all or even most of the difference in speeds between these two. It also looks like the real cells still have a rather messier oscillation, right? You can see like they don't go back to baseline the way the simulated repressor really always goes back to. So we might ask, all right, can we tune this simulation to maybe make it messier to more realistically capture what we see in the lab? And if you remember, one of the ways we can break the oscillator is by turning up or down decast production. So let's try that. This is showing simulation results with decast9 tuned down from right to left. And what you see is that, yeah, eventually you break the oscillator so it becomes basically just noise, where it's not really clear that there's any coherent or systematic oscillation. But I would say that these aren't particularly predictive looking examples. I don't think we get particularly close to the real CRISPR. The picture's a little different with data from the other paper. This is data from the Paulson lab. And Paulson's group made a CRISPR on a low copy plasmid, which does turn out to make some difference. If we simulate the CRISPR on a low copy plasmid instead of medium copy, and then we tune down decast production, we actually get some results that look kind of startlingly close to the real results. So I would claim that somewhere in this range, we found a sweet spot that maybe accurately represents the noisiness that you get in a real CRISPR. And now, with our model, we have some potential interventions that could be added by an entrepreneurial experimentalist to try to turn this into something more like the smooth, regular oscillations that we see in other simulations. So I'm out of time to talk about the CRISPR later, But I do just want to mention that the same model can be used to investigate and describe a number of other CRISPRi circuits. You can make toggles, you can make pulse generators that make a temporary pulse. You can hook these things together and make multi-pulse generators driven by a CRISPRilator. And it looks like CRISPRi can actually do interesting things, and it has scaling limits that are important, and you can find those scaling limits through simulation. But it is perhaps worth pursuing, and interestingly, the CRISPRilator in particular may be a kind of borderline case where it is uniquely right on the edge of functionality. So in conclusion, CRISPRi looks like it is promising, but it has limitations and we need to know about them. And really it makes sense to investigate those perhaps before spending a lot of time, effort, and money in the lab building them. CRISPRilators in particular are right on the edge of functioning. They work, but barely, and can probably be improved with some specific targeted changes that I've recommended, like tuning decast production rate and adding small amounts of protein degradation. And finally, we have a model that can let us investigate a wide variety of CRISPRi-based synthetic circuits and can, again, determine whether or not they're worth building in the lab and what we might have to do to make them work in the lab. So I have many people to thank for all of the work that I've been able to do. And first and foremost among those by far is Richard Murray. Richard, as both an advisor and maybe more importantly as a manager, you have done a fantastic job of making your lab a good place that we can safely explore new science, try interesting things. It's been a fantastic place to work, and I thank you for it. And I would be remiss not to mention my funders, who also made all this work possible. I also would like to thank the other members of my committee. So Dr. Eric Winfrey has worked with me since I started here. And in particular, I need to thank him for bringing me into his surface chemical reaction network work that forms the basis for my first thesis chapter, which was not shown here for lack of time. Leah and Paul have been there for me also pretty much since the beginning. Thank you both for inspiring me and for consistently helping me find the right questions to ask in my research, and sometimes even helping me find the answers to those. Not on my committee, but I also want to thank Dr. Liu Qian again for her work bringing me in on the Surface CRN project, but also as a general inspiration for me and I think a number of my colleagues in bioengineering. I want to thank our lab manager, Mickey Yun, for helping me navigate the graduate student and academic experience, and for a number of really valuable conversations had over Hongzhou and homemade desserts. I want to give a special shout out to Justin Boyce, who has been single-handedly responsible for really training an entire generation of biology and bioengineering students here at Caltech. Justin is responsible for a huge shared toolkit of ideas and techniques and tools that has really shaped how we've done research and how we've approached scientific questions. This is not an exhaustive list. This is a semi-exhaustive list of the many people in the lab who have made the Murray Lab a fun, sane, and fascinating place to do science. So thank you all. And especially I thank these four: Kevin, Jean, Andre, and Eric Ju. These are my housemates, present and prior, who have consistently made my home a welcoming and educational place to be, which has been especially important this last year and a half, roughly. You guys have made a big difference to my graduate student experience, and you have a lot to teach the world. I'm glad I've been able to absorb a little bit of that. Very personal note, I want to thank John and Andy and Caroline, who have been with me in my academic voyage for quite a while now. I would not be the person I am without the three of you, much less where I am doing what I am. Also, special little thanks to John Markin for helping me personally hammer out this particular presentation and forge it into what it is now. And finally, I want to thank my family. So my parents have literally made everything I've done possible. huge thanks to them. And also to my wife, Sarah, for keeping my head on straight, facing in the right direction, and keeping my feet moving forward for many years now. And thank you all for taking the time out of your day to hear a little bit about CRISPRi systems and what they can do and what they can't. And I hope you learned something. So
Fantastic.
Yeah, I can move to questions.
I will clap on behalf of everybody, but you can do your visual claps as well. So you can pretend like that got magnified by, sort of 50 or so. Okay, so we do have a little bit of time for questions from the general group before we go off and have the committee grill Sam. But if there are questions from the public, feel free. Maybe raise your hand, or you can also just try and jump in as you prefer.
Okay, if we're gonna raise hands, I should look at the gallery view. Yeah?
I can look at it. I'm looking at it.
Okay, yeah.
Deanna.
Hi, Sam. Great talk.
Thank you.
I was wondering, early on, you mentioned that gRNA circuits can't have cooperativity. I was wondering if you had binding sites within different gRNAs, if you could induce cooperativity and what effect that would have on your circuits.
If you could induce cooperativity by having multiple, are you thinking like multiple Cas9s attached to a single guide RNA?
Yeah, or different guide RNAs that when they bind, they only expose like one binding site, for example.
That's an interesting question. So I'm going to take a bit of a cop-out answer and reference eukaryotic CRISPRi. So everything I've shown is for prokaryotic CRISPRi. Eukaryotic CRISPRi is a little different and typically works by fusing dCas9 to some other repressor and replacing the binding portion of that other repressor with dCas. Right, many of those repressors are cooperative. I would not be shocked if you could get cooperativity with some of those. So it may be possible to get cooperativity. Importantly, I don't think you have to, but it may be possible, yes.
Thank you.
Okay, other questions for Sam? Sorry, I'm adjusting my light here a little bit. V10, is that Chewy? I can't tell who that is.
Yeah, it's me. Hey Sam, like your presentation.
Thank you.
I got a question. So I noticed like one part you said if the binding was too strong, you couldn't really get the repressor to work. I was wondering, why haven't you tried, did you try like the spy Cas9 where they did mutations to make it more accurate, but the binding was weaker? As well as what happens if you just switch it to an activator Cas9 in a prokaryotic system? Would that make it work better?
Right. Okay. So two questions there. One was, have we looked at other engineered variants of Cas9 that are weaker binding but more accurate? And is that right? Is that correct? That the question? Yeah. Okay. And second question, have we looked at CRISPR activators where binding turns on transcription? To answer the first one, this model is actually probably not sufficiently complex to change that. We don't actually model off-target binding of dCas9 by default, but we can add it. And what you see is that as you add more and more off-target binding, it slowly kills the repressor. So I think that may be a little outside the scope of the simulations I've done. As for activators, I have done some work with CRISPR activators. The cool thing about CRISPR activators is that they scale better in the sense that they are not as affected by guide competition. So if you add, as you add guide RNAs, as I've shown, CRISPR inhibition drops off very, very fast. CRISPR activators should not drop off nearly as fast. So if you can do things with activators, you should be able to squeeze more of them into a cell before they break. There are some things you can't build with CRISPR activators that you can with repressors, so there's some limits to that. But I hope that answers your question. I would refer you to my thesis, I guess, for the details on that.
Okay, Harriet.
Hi, I have a question. So I see all of your work is it's been done in the E. coli. I was wondering, can this system actually functional in the mammalian cell?
Yes, the question is, we've done all this in E. coli. Does the same system work in eukaryotes like humans? And the answer is sort of. This exact system doesn't. It turns out if you try to throw this exact DCAS9 system into eukaryotes, it just falls off immediately. It doesn't actually block eukaryotic transcription. And we think that's because eukaryotic polymerases are really aggressive and will just displace this repressor. You need something stronger.
Yeah, and also like the, I don't know whether it's true, but I think most of the repressor is for like long transcript, but like the guide RNA is on the promoter for like, sorry, it's on the different kind of polymerase than those long transcripts. So I don't exactly know whether those repressor can work on the short RNA transcript.
Right. So, oh yeah. So you're asking, can you actually use a eukaryotic CRISPRi to block transcription of guide RNA?
Right.
Yeah. Well, you can always make a transcript longer. You can always put self-splicing domains in a long transcript. So you have to make like a kill base before you get to the guide RNA.
Yeah.
And again, there are eukaryotic, just for the audience, there are eukaryotic CRISPR repressors. They work a little differently. They're essentially existing eukaryotic repressors with the binding domain replaced by a Cas9 binding domain.
Okay, thank you.
Yep, thank you.
Great. Okay, well, we've hit kind of the top of the hour. So let's thank Sam again for a great seminar or thesis defense. And with that, I'll ask everybody except the committee to bid your farewells by leaving the meeting, and the committee will stick around. Thanks everybody for coming. Sam, you can probably turn off the recording.
Probably should.